Welcome to the Stay Curious channel. Today, we will talk about oxidative phosphorylation, how cells produce ATP. Let's begin. Every cell in the body requires energy to survive, grow, repair itself, and perform its normal functions. Most of this usable cellular energy is stored in a molecule called adenosine triphosphate, commonly known as ATP. One of the most important processes responsible for producing ATP is oxidative phosphorylation. In eukaryotic cells, oxidative phosphorylation takes place inside the mitochondria. Mitochondria are often called the powerhouses of the cell because they convert energy from nutrients into ATP. A mitochondrion has an outer membrane and a highly folded inner membrane. The folds of the inner membrane are called cristae. These folds increase the available surface area for the proteins and enzymes involved in ATP production. The space enclosed by the inner membrane is called the mitochondrial matrix, while the narrow region between the inner and outer membranes is called the intermembrane space. Oxidative phosphorylation consists of two closely connected processes, the electron transport chain and chemiosmosis. The electron transport chain is a series of protein complexes located within the inner mitochondrial membrane.
These protein complexes transfer electrons from one carrier to another. The electrons entering the chain are mainly supplied by NADH and FADH₂. NADH and FADH₂ are produced during earlier metabolic pathways, including glycolysis, pyruvate oxidation, the citric acid cycle, and fatty acid oxidation. These molecules carry high-energy electrons that were originally obtained from nutrients, such as carbohydrates and fats. NADH transfers its electrons to complex 1 of the electron transport chain. As the electrons move through complex 1, energy is released. Complex 1 uses this energy to pump hydrogen ions, also called protons, from the mitochondrial matrix into the intermembrane space. The electrons are then transferred from complex 1 to ubiquinone, also called coenzyme Q. FADH₂ enters the electron transport chain through complex 2. Unlike complex 1, complex 2 does not pump protons across the membrane. However, it transfers electrons from FADH₂ to coenzyme Q. Coenzyme Q carries electrons from complex 1 and complex 2 to complex 3. As the electrons pass through complex 3, more energy is released. Complex 3 uses this energy to pump additional protons from the matrix into the intermembrane space. The electrons are then transferred to cytochrome C. Cytochrome C is a small, mobile protein located near the outer surface of the inner mitochondrial membrane. It carries electrons one at a time from complex 3 to complex 4. Complex 4 is the final protein complex in the electron transport chain. At complex 4, the electrons are transferred to oxygen. Oxygen acts as the final electron acceptor. The oxygen combines with electrons and protons to form water. Without oxygen, the electron transport chain would stop because the electrons would have nowhere to go. As electrons move through complexes 1, 3,
Complexes 3 and 4, protons are continuously pumped from the matrix into the intermembrane space. This creates a high concentration of positively charged protons in the intermembrane space and a lower proton concentration in the matrix. The unequal distribution of protons produces an electrochemical gradient across the inner mitochondrial membrane. This gradient is called the proton motive force. It contains stored potential energy, similar to water being held behind a dam. The inner mitochondrial membrane is largely impermeable to protons. Therefore, the protons cannot simply diffuse back into the matrix through the membrane. Instead, they return through a specialized protein called ATP synthase. ATP synthase functions like a molecular turbine. As protons flow through the membrane portion of ATP synthase, they cause parts of the enzyme to rotate. This rotation changes the shape of the catalytic regions of ATP synthase. The enzyme then combines adenosine diphosphate, or ADP, with inorganic phosphate to produce ATP. The movement of protons through ATP synthase and the resulting production of ATP
is called chemiosmosis. The electron transport chain creates the proton gradient, while ATP synthase uses the energy stored in that gradient to manufacture ATP. Together, these two processes make up oxidative phosphorylation. Oxidation refers to the transfer of electrons from NADH and FADH₂ through the electron transport chain. Phosphorylation refers to the addition of inorganic phosphate to ATP.
Oxidative phosphorylation produces most of the ATP generated during aerobic cellular respiration. The oxidation of one NADH molecule generally supports the production of approximately 2.5 ATP molecules. The oxidation of one FADH₂ molecule generally supports the production of approximately 1.5 ATP molecules. FADH₂ produces less ATP because its electrons enter the electron transport chain through complex 2, bypassing the proton pumping activity of complex 1. Oxidative phosphorylation is not perfectly efficient. Some of the energy from the proton gradient is released as heat. A small number of electrons may also escape from the electron transport chain and react prematurely with oxygen. This can produce reactive oxygen species. At controlled levels, reactive oxygen species can participate in cellular signaling. However, excessive production can damage proteins, lipids, mitochondrial membranes, and DNA. Cells therefore contain antioxidant defense systems that help regulate reactive oxygen species and protect mitochondrial function. Certain chemicals can also interfere with oxidative phosphorylation. Electron transport chain inhibitors block electron movement through specific protein complexes. For example, rotenone inhibits complex 1, antimycin A inhibits complex 3, and cyanide inhibits complex 4. Oligomycin inhibits ATP synthase and prevents protons from flowing through the enzyme. Uncoupling agents act differently. They allow protons to return to the mitochondrial matrix without passing through ATP synthase. This increases oxygen consumption and heat production, but reduces ATP synthesis. In summary, oxidative phosphorylation begins when NADH and FADH₂ donate high-energy electrons to the electron transport chain. The movement of electrons through the chain releases energy. This energy pumps protons into the intermembrane space and creates a proton gradient. The protons then flow back into the mitochondrial matrix through ATP synthase. ATP synthase uses this flow to convert ADP, an inorganic phosphate, into ATP. Finally, oxygen accepts the electrons and combines with protons to form water. Through this coordinated process, mitochondria convert energy from nutrients into the ATP that powers cellular life. Don't forget to share, like, comment, and subscribe. Stay curious.